to the BCC19 COVID-19 test tutorial from Mobile Detect Bio. This tutorial is only for the FDA EUA authorized BCC19 test available on mobiledetectbio.com and is not for use with any other product. In this video, we will be going over how to accurately perform the BCC19 test as well as analyze its results. The BCC19 test can be broken down into 4 easy steps. We have sample collection. Then, we will prepare the test reagents. After that, we add our sample. Finally, we run the test and analyze its results. Now, let's take a closer look at each step. Step 1. Sample collection. The test uses one of the following swabs for sample collection: anterior nares or nasopharyngeal. For storage and transport of the swab, the test uses saline or viral transport medium. The person collecting the sample should follow protocols outlined by the CDC for safety, such as wearing the appropriate PPE. When collecting the sample, take care not to touch the end of the swab to yourself or other surfaces. When swabbing the patient, please follow CDC guidelines for swabbing technique. Once you have collected the patient's sample, insert the swab tip first into the transport tube. Bend and break the swab on the perforation to remove the excess swab stem. Make sure the cap of the transport tube is sealed and secure. This excess stem should be disposed of following state and local guidelines for biohazard waste. Samples can be stored for up to 72 hours at 2 to 8 degrees Celsius. For storage times longer than 72 hours, store the sample at or below -70 degrees Celsius. Step 2. Prepare the test reagents. Reagent preparation should be in a sterile setting separate from the sample collection area. The best area to work in is a PCR hood or clean workstation. Proper cleaning procedures are important to consistently running a successful test. The 2 cleaning solutions should be a 10% bleach solution as well as at least a 70% alcohol-based solution. Clean with the bleach solution first, followed by the alcohol solution, both before and after testing to avoid contamination. Also, be sure to clean your pipettes and stands regularly. When prepping the reagents, a mask, gloves, and lab coat should be worn. If not using sterile gloves, the gloves should also be cleaned with the alcohol-based solution. Reagents 1 and 2 should be thawed all the way through before using them. It is best to thaw the reagents on ice. Remove a PCR strip from its bag, then place the PCR strip into the MD Bio preparation stand. Also, place a lid strip with the openings upward either onto the MD Bio preparation stand or a cleaned area of your workstation in order to avoid contamination. Before adding reagent 1, invert it 5 times to ensure an even mixture. Repeat the same process with reagent 2. Alternatively, the reagents can be vortexed. Using a pipette, adjust the volume amount to 12.5 microliters. Attach a sterile, filtered tip to the pipette. In accordance with the number of tests that you are running, add 12.5 microliters of reagent 1 to each well of the PCR strip. Once reagent 1 is added to all desired wells, dispose of the pipette tip and close the reagent tube. When preparing reagent 2, adjust the volume amount of the pipette to 11.5 microliters. Attach a new pipette tip. Now add 11.5 microliters of reagent 2 to your test wells. Since reagent 1 is already present in the test well, use a new pipette tip for each reagent 2 suspension. Doing so reduces the risk of reagent contamination and crossover. Note that it's important that reagent 2 gets all the way down in the well and combines with reagent 1. When all wells have been prepared, close the reagent 2 tube and then place the PCR strip lid back onto the PCR strip. For testing volumes of 8 or greater, it is recommended to use the aliquoting method. With this method, we are mixing the reagents together before adding them to the PCR wells. For reagent 1, adjust your pipette to 102 microliters. Add reagent 1 to a separate sterile tube. After disposing the tip, adjust the pipette to 94 microliters for reagent 2. Add reagent 2 to the same tube. To add the aliquoted reagents into the PCR tubes, adjust the pipette down to 24 microliters. Lightly agitate the mixture. Add 24 microliters per well. Because we aliquoted, only one pipette tip is needed for all 8 wells. When all reagents have been added, close up the PCR strip. Store reagents 1 and 2 on ice in between tests. If there is greater than 8 hours in between testing, then store the reagents at -20 degrees Celsius. Step 3. Sample addition. For sample addition, a separate work area must be used that has also been cleaned and sterilized. Place the PCR strip into a separate MD Bio preparation stand and remove the lid. Set the pipette to 1 microliter for sample addition. Agitate the sample by inverting 5 times. You can also vortex the sample. With a filtered pipette tip, add 1 microliter of sample into a single well. If the cap has visible fluid in it, pull the sample from the cap for cleanliness. Make sure to plunge the pipette tip into the liquid before dispensing. This will ensure the sample mixes with the reagents. Alternatively, you can pipette the sample from the bottom of the transfer tube. When using this method, the pipette must be cleaned with alcohol between each sample. To validate the reagents and test cycle, it is recommended to do 1 positive control and 1 negative control per heating cycle. Like the reagents, the positive control should be thawed on ice before use. Once thawed completely, invert the positive control 5 times. You can also vortex the positive control. With the pipette set to 1 microliter, add the positive control to a single well. Just like the sample, make sure that the pipette tip is in the liquid before dispensing. To run a negative control, add 1 microliter of clean transport medium or saline to its own separate well. When all samples and controls have been added back to the wells, place the lid back on the strip. Store the BCC19 positive control on ice between tests. If the delay between testing is greater than 8 hours, store the positive control at -20 degrees Celsius. To avoid contamination, store the positive control separately from the reagents. To ensure that the fluid gets to the bottom of the wells, hold the PCR strip in the middle and flick your wrist. Instead, you can centrifuge the strip. Step 4. Run test and analyze results. We will now go over how to incubate the test on a 96, 32, and 8 well thermocycler. For the 96 well heater, insert the test for incubation. Follow the on-screen prompts to start your incubation cycle. Remove the test strip when the cycle is complete. The incubation process for the 32 well heater is similar. Insert the tests, follow the on-screen prompts to run the incubation, and then remove the tests when they're complete. For the 8 well heater, insert the tests, close the lid, and press the select button to start the test cycle. The ready and testing lights will flash when the testing cycle is concluded. Press select to return to standby mode. Remove your tests and proceed to analysis. For the 8 well heater, the strip must be removed within 5 minutes of the end of the test cycle, or that strip will be considered invalid. The BCC19 test results can be analyzed visually by interpreting the colors. Red or pink indicates a negative result, whereas yellow indicates a positive result. In addition to visual test analysis, you have the option of utilizing the free Mobile Detect app, available for iOS and Android. Place the tests into the MD Bio analysis stand. Using the Mobile Detect Bio app, scan the QR code on the analysis stand. The app will analyze the results and create a digital report. The app can then be used to log patient data, as well as generate and send reports. That concludes this tutorial for Mobile Detect Bio's BCC19 COVID-19 test kit. If you have additional questions or concerns, please visit mobiledetectbio.com for more information.